Welcome back, guys. We are discussing about the muscle physiology, how the muscles are going to be contracted. Now in this part 2 of the muscle physiology, first let's have a recap and let's see more important points about the muscle contraction. Okay, having said that, now first let's recap the things, okay, what we have discussed so far. Now I have shown you how the muscle cell membrane, okay, the cell membrane, the sarcolemma is forming the invaginations into the cytoplasm. What are these invaginations called as? So these invaginations which I am showing you here, you know it by this time already, these are called as T-tubules, okay, they are called as T-tubule, okay. Now between these T-tubules there are these L-tubules present. Now can you tell me what exactly are these L-tubules? The L-tubules, L for longitudinal, these L-tubules are nothing but the sarcoendoplasmic reticulum, which contains high amounts of calcium in this last region called as cisterna region. In this last cisterna region, which are called the cisternae, lots and lots of calcium is stored, okay. Now look here, this is the skeletal muscle, okay. Here is this is the skeletal muscle, okay. Now let me first discuss what exactly happens in the skeletal muscle, okay, you know it. Now when a alpha motor neuron, okay, when an alpha motor neuron, let me show you here this motor neuron in the green color, this is the motor neuron. This motor neuron, what it will do? It will release the acetylcholine. Now this acetylcholine is going to act on its receptors, so that what happens is the sodium will try to come into the cell, okay. There will be sodium influx, okay, sodium is coming into the cell. So sodium is coming into the cell, so this sodium is responsible for the depolarization of this membrane. When the muscle membrane is getting depolarized in a skeletal muscle, what exactly is going to happen? This depolarization is passing on the membrane of the skeletal muscle, on the sarcolemma. So who is sitting on the T-tubule? You know, there is a channel who is sitting on this T-tubule, okay. There is this channel which I am showing in the green color. So this channel is going to get stimulated. So because of this depolarizations, now what happens is this green color channel is going to get activated. So what is the name of this green color channel? Already I have explained you. This green color channel is nothing but DHPR, so dihydropyridine receptor calcium channel on the skeletal muscle. This is the skeletal muscle membrane, right? So it is called as CAV 1.1. So this is a voltage gated calcium channel 1.1. Okay, it's a voltage gated calcium channel. So it's coming under L type of calcium channel. It's a type of L type of calcium channel. There are different types of calcium channels. Okay, there are different types of calcium channels. L type, okay, P type, N type, okay, T type. There are different types of calcium channels. The point which I want you to know is on the skeletal muscle, okay, on this T-tubule, there is a type of L, there is a L type, okay, there is a L type calcium channel that is present, that is called as CAV 1.1 or DHPR, dihydropyridine receptor calcium channel, okay. So here let me give you a list so that it will be helpful for your exams. Calcium channels, okay. What are the different types of calcium channels are there? The first type of calcium channel just now I have discussed with you, L type of calcium channel. These L type of calcium channels are also called as dihydropyridine receptor calcium channels. Dihydropyridine receptor calcium channels, okay, DHPRs. Now it's of two types. CAV 1.1, it is present where? CAV 1.1 is present on skeletal muscles, okay. CAV 1.2 type is present on myocardium as well as smooth muscles, okay. So this is a first type of calcium channel, L type of calcium channel, standing for the long standing calcium channels. Next type of calcium channel is called as P type or Q type. So P type or Q type, they are CAV 2.1. Okay, CAV 1.1, 1.2 completed. Now this is a different type of calcium channel which is called as a voltage gated calcium channel 2.1. It is present on neurons. Next, N type. Okay, N type neuronal CAV 2.2. These are also present on the neurons. So on the neurons, which type of calcium channels are present? P type or N type. Okay, so P type, N type calcium channels, they are present on the neurons. So how many completed? L type completed, P type completed, N type completed. Now the next type is called as the T type calcium channels. So this T type transient calcium channels CAV 3.1, and different types are there, 3.1, 3.2, 3.3, something like this. So these CAV 3.1, 0.2, 0.3, okay, 3.1, 3.2, 3.3. Where exactly are they present? We have discussed this even in the cardiovascular physiology on the pacemaker cells. Okay, there are T type calcium channels, there are L type calcium channels. There also we have discussed. So it is present on the pacemaker cells, that's a SA node, AV node, and the pacemaker cells, pacemaker cells and osteocytes. So on these areas you are having this T type of calcium channels. Okay, so finally our topic is this skeletal muscles. Okay, on the skeletal muscles, which type of calcium channels are present? L type. L type are also called as DHPR. So which type, which subtype? CAV 1.1. Okay, so this one, CAV 1.1 type is present. Now what happens? Now look, as the depolarization is spreading, okay, as the depolarization is spreading, see the membrane is getting depolarized, later it will become repolarized, depolarization. The action potentials are happening on the muscle membrane. So ultimately this DHPR calcium channel is going to open. When the DHPR calcium channel opens, I have already explained you in the last video, it opens, it mechanically opens. Okay, it mechanically opens one more calcium channel which is present on this L-tubule. This is L-tubule, you know it. So on the L-tubule there is this one more calcium channel which is present in this blue color, that will be mechanically opened. So this DHPR channel, once it is electrically stimulated, it will come and it will open the RYR calcium channel. So the blue color channel which I am showing here is called as a ryanodine receptor calcium channel. So this fellow name is RYR type 1. Okay, so when the RYR type 1 is opened, immediately what happened? The calcium whatever is stored inside this sarcoplasmic reticulum, this calcium is going to come out. This calcium will be available for the muscle contraction. What that calcium will do, that we can discuss later. So what I have explained you so far. So because of the excitation of the muscle membrane, the action potentials are going on the cell membrane of the muscle, the T-tubules are getting depolarized. Because of that, the DHPR channel mechanically opens the RYR channel type 1. Okay, RYR is a calcium channel. Okay, so let me write here a list RYR channels. Okay, let me write RYR channels. Okay, just now I have explained you where exactly is RYR type 1 present. The RYR type 1 is present on RYR type 1 is present on skeletal muscles, present on the skeletal muscles. RYR type 2 is present on myocardium, that's the heart, and RYR type 3, it is present on smooth muscles. Okay, so these are the different types of ryanodine receptors. Okay, now what else you should know for your exam is, sir, on the skeletal muscles, which type of ryanodine calcium channels are present? RYR type 1. So now these RYR channels will be opened mechanically with the help of DHPR channels. So the calcium which is present in the sarcoplasmic reticulum, this calcium is going to come out into the cytoplasm, and that calcium will be helping in muscle contraction that we will discuss later. Now questions. Okay, now questions. See, this sodium channel which I am showing here. Okay, this is sodium channel which I have shown here. See, the sodium channel name. What is the name of this sodium channel? So, what is the name of this sodium channel? It's a voltage gated sodium channel. So, this voltage gated sodium channel is called as Na sodium voltage gated channel 1.4. So, voltage gated sodium channel 1.4. Okay, it's the name of that sodium channel. Okay. See, here I have explained you different types of calcium channels. Also, I have explained you different types of RYR channels and their location. Let me explain you different types of voltage gated sodium channels and their locations so that you will having a better understanding. Now, let me write here sodium channels. Okay, voltage gated sodium channels. There are 1.1 to 1.9. Okay, there are different types. Okay, there are different types. Our topic is about the skeletal muscle. Okay, skeletal muscles. On the skeletal muscles, which type is present? Okay, NAV 1.4 type of voltage gated sodium channel. It's present on the skeletal muscles. Okay, at the neuromuscular junction. Next, on the myocardium. On myocardium, voltage gated sodium channel 1.5 type is present. And all the others, okay, and all the others, okay, all the other voltage gated sodium channels except 1.4 and except 1.5, they are present on the neurons. So now we are discussing about the skeletal muscle. So on the skeletal muscle, which kind of a sodium channel present? Voltage gated sodium channel number 1.4. Okay, based on the subunits, they are classified into 1.1, 1.2, 1.3, 1.4, 1.5, something like this. So on the skeletal muscles, which type of voltage gated sodium channel present? Voltage gated sodium channel 1.4. Now if it is a mutated MCQ, if it is a mutated, it's going to cause a condition called as hyperkalemic. Okay, it's going to cause a condition called as, let me write here itself. Okay, if it is a mutated, it cause hyperkalemic, okay, periodic paralysis. Okay, so hyperkalemic periodic paralysis is going to be seen as a result of mutation of voltage gated sodium channels. Now, what if this channel is mutated? Okay, this DHPR channel is mutated. In the last video, I have already explained you, if this DHPR channel is mutated, okay, if this DHPR channel, if it is mutated, it is going to cause a condition called as hypokalemic periodic paralysis. Okay, so hypokalemic periodic paralysis is because of the mutation of DHPR channel. Hyperkalemic periodic paralysis is due to mutation of the voltage gated sodium channel. Now one more channel is there. What is this one? RYR1 channel. Okay, RYR1 channel. Now if this RYR1 channel, if it is mutated, there is gain of function mutation. Okay, if there is gain of function, of mutation of RYR channel type 1, that's going to cause a condition called as, I have already explained you in the last video, that is called as malignant hyperthermia. Malignant hyperthermia. Okay, where the muscles are contracting so much, the RYR channels are kept on opening for long duration of time. So muscles are contracting for so long time, so that there will be more temperature production. That's why hyperthermia, malignant hyperthermia. Okay, so when the muscles are contracting for so much time, the muscle fibers can be damaged. So that will cause rhabdomyolysis. Rhabdomyo means skeletal muscle is getting broken down. So that's going to cause a rhabdomyolysis. Okay, so in malignant hypothermia, the patient is going to develop rhabdomyolysis. Because of this rhabdomyolysis, myoglobin is going to leak into the kidneys, and that will cause acute kidney injury. Okay, acute kidney injury. That's integration with the pathon medicine. Now, what if this RYR channel is losing? Okay, if this RYR channel Type 1, there is loss of function. Okay, hypofunctioning, it's not functioning. That's going to cause a disease called as central core diseases. Central core diseases. So these are the points which I want you to know. Central core diseases are due to loss of function mutation. Gain of function mutation is going to cause a malignant hyperthermia. Usually this malignant hyperthermia, it is going to be associated with your anesthesia. Which people develops this malignant hyperthermia? You know, gain of function mutation. But when these people, when they do excessive exercise, what are the precipitating events? Okay, certain anesthetic agents, and during excessive exercise they are going to precipitate this malignant hyperthermia. What I want you to know for your exams is drugs like halothane, 𝗠𝐶𝐼𝐿-CONIN, okay, sevoflurane. So these anesthetic agents, okay, inhalational anesthetic agents, and like certain muscle relaxants like 𝗠𝐶𝐼𝐿-CONIN, they are going to precipitate this event called as malignant hyperthermia. So there is excessive muscle contraction, and I have also explained what is the treatment for this malignant hyperthermia in the last video itself. What is the treatment? The treatment should be the blocker of RYR channels. The RYR channels are doing here gain of function, they are doing overaction, they are doing more function. So what you have to do, you have to block them. So the drug is dantrolene sodium. Okay, that drug is dantrolene sodium. I have already explained you. Now here, what else I want you to know is, in the skeletal muscle contraction, we are in the beginning. Okay, we are in the beginning. We haven't seen so far how the actin and myosin are interacting. We haven't seen all that story. Here, what and all I have explained you, the entire concept is called as electromechanical coupling. Okay, what exactly is this event called as? Electromechanical coupling. So what exactly is this electromechanical coupling? See, when the muscle membrane is getting electrically depolarized, the RYR channel will be opened. Okay, when the muscle membrane is getting electrically depolarized, the first event electro. So what is this word mechanical means? Mechanical stands for first the DHPR channel, now it will go and open the RYR channel mechanically, so that the calcium will be available for the muscle contraction. So this event is called as electromechanical coupling. Okay, next thing, in the skeletal muscle contraction, okay, in the skeletal muscle contraction, now say imagine this muscle is contracting. So in how many ways the calcium is coming into the cell? So how many sources of calcium are there? There is only one source of calcium. So in skeletal muscle contraction, let me write here, in skeletal muscle contraction, there is only one source of calcium. Okay, there is only one source of calcium, and what is the one and only one source? That is sarcoplasmic reticulum. Okay, sarcoplasmic reticulum. Now what is something different? Okay, what is something different from the myocardium? Okay, you already know it. See, if this is the myocardial cell, this is the myocyte. Now here I am showing you what is this one. Now this is the T-tubule, you know it. Now attaching to the T-tubule, very close to the T-tubule, what is there? This L-tubule is there. Okay, here is the L-tubule. Okay, now on the myocardium, you should tell me what is this channel present. This channel which I am showing here, this channel is called as DHPR channel. Okay, so this channel is DHPR again, but which kind of DHPR? See, in the skeletal muscles, I have told you there will be, look here, in the skeletal muscles it will be CAV 1.1, but on the myocardium, what is the type of DHPR channel present? That is CAV 1.2, voltage gated calcium channel. Okay, CAV 1.2, it's a CAV 1.2. Okay, now here what is this channel which I am showing? So this channel which I am showing here, this is RYR channel. But what kind of RYR channel? On the myocardial cells there is RYR type 2, RYR type 1. Already let me show you here. RYR type 1 is present on the skeletal muscles, RYR 2 is present on myocardium. Okay, so what exactly happens when the muscle membrane is getting depolarized? Okay, when the muscle membrane is getting depolarized, the depolarization is sweeping. Okay, it's sweeping on the T-tubule. Now what happens for the first time? Look, what happens for the first time is, now when the depolarization is coming towards this DHPR channel from outside, from the extracellular fluid, first calcium will come into the cell. Okay, first the calcium from outside is coming into the cell. Now what this calcium is going to do, I have explained you, this calcium is going to open the RYR channel. It's not the mechanical opening. Okay, so this DHPR channel and RYR channel, they are interacting. Okay, so what I have explained you here, the calcium which came into the cell, is allowing the RYR channel to release the calcium. Now the calcium, whatever is there in this sarcoplasmic reticulum, so this calcium will be released out into the cytoplasm. So now tell me, in the myocardial cell, how many sources of calcium are there? Again, the contraction of the myocardial cell, how many calcium are there? There are 2 sources of calcium. Let me write it here. 2 sources of calcium. So one calcium is coming from the extracellular fluid with the help of DHPR channel, and this calcium is stimulating the sarcoendoplasmic reticulum to release the calcium through the RYR channel. So it's the first calcium which is important calcium. It's the first calcium which comes into the cell. It stimulates the RYR channel to release the calcium. So this first calcium is called as essential calcium. Okay, so the first calcium which came into the cell is called as the essential calcium. Okay, so it's the extracellular calcium which came into the cell and stimulating the release of intracellular calcium. So this event is called as calcium induced calcium release. So it's the outside calcium which is coming in and stimulating the release of intracellular calcium. So this event is called as calcium induced calcium release. Okay, calcium induced calcium release. So this is the point which I want you to know. There are 2 sources of calcium. One is essential calcium, and there is something calcium induced calcium release phenomena which is seen in myocardial cell. Now one more thing which I want you to know here is, in the skeletal muscles, okay, in the skeletal muscles there is electromechanical coupling. It's the excitation of the muscle cell membrane which is causing the mechanical opening of the RYR channels. The DHPR channels are mechanically opening the RYR channels. So that is called as the electromechanical coupling. Now here in the myocardial cells, it is little different. This is electrochemical coupling. Okay, because of the excitation, the calcium is coming and helping in the release of the calcium. So this event is called as chemical coupling. So tell me, electromechanical coupling is happening in the skeletal muscle. Okay, electromechanical coupling is happening in the skeletal muscle, and electrochemical coupling is happening in the myocardium. So both are different. In myocardium there are 2 sources of calcium. In the skeletal muscle there is only 1 source of calcium. There the channels are different. There the sodium channel, there the DHPR channel, and there the calcium channel are different. Here the sodium channels, the calcium channels, and the RYR channel, okay, DHPR channel and the RYR channel, they are different. Okay, so these are the basic things which I want you to know. Now later, what else I should know, I want you to know is, see here, again let me draw. This is your sarcolemma with the T-tubule. Here is the sarcoplasmic reticulum which contains lots and lots of calcium. Okay, now calcium came out finally. Okay, finally calcium came out of the sarcoplasmic reticulum because of the electromechanical coupling. I am talking about the skeletal muscle. Okay, first let me talk about the skeletal muscle. Because of electromechanical coupling, calcium came, and calcium is going to help in the muscle contraction. That we will discuss later. How the actin, myosin, the sliding filament theory, that we will discuss later. Now the calcium came out after the muscle contraction. Where this calcium should go? I have already explained you. This calcium, it should go back. Okay, it should go back into this sarcoplasmic reticulum with the help of this pump. With the help of a pump called as Sarca. Okay, Sarca. So sarcoendoplasmic reticulum calcium ATPase. So what it is doing? It is sequestering the calcium back into the endoplasmic reticulum. Okay, so it's trying to decrease the intracellular calcium levels so that there won't be any inappropriate muscle contraction. So the Sarca is concentrating the calcium from low concentration to high concentration, because in the cell there is very little calcium, but within the sarcoendoplasmic reticulum there is lots and lots of calcium which is present over there. So calcium is again trying to go back. Okay, so from low concentration to high concentration, low to high concentration. So this Sarca is an example of primary active transport. It's an example of primary active transport. Now this Sarca is trying to help in relaxation. Okay, it's trying to help in relaxation because it is taking the calcium back in. So helping in the muscle relaxation. Now where all this calcium is going? Tell me. Sir, this calcium is trying to go back into the sarcoendoplasmic reticulum. The calcium is trying to come and store here. Now imagine if here in the sarcoendoplasmic reticulum, okay, in the sarcoendoplasmic reticulum, if you have increased the calcium concentration so much, it will be difficult for this Sarca pump to get the calcium back into the endoplasmic reticulum. Of course, it is using ATP, of course, it is using energy. But if you increase the calcium levels here in the endoplasmic reticulum so much, it will become difficult for the Sarca pump to sequester the calcium. Okay, so for that purpose, what exactly happens is, inside this sarcoplasmic reticulum, there are proteins present. These proteins, what they will do is, they will bind. Okay, there are proteins which will bind with this calcium so that the calcium will not be free. Okay, so what I am trying to put into your mind, inside the sarcoplasmic reticulum, there are proteins present. These proteins, they will go and bind with the calcium so that the bound form, the free forms are going to contribute to, like, the concentration gradients. But once the calcium is bound, means it's not going to contribute any to any concentration gradient. So what are these proteins which are present in the sarcoplasmic reticulum? The first one is called as calsequestrin. This calsequestrin, it is present in skeletal muscles, myocardium and smooth muscle. Okay, it's present in also skeletal muscles, myocardium as well as smooth muscles. One more protein called as the 𝗠𝐴𝐿-reticulin. This is also calcium binding protein. So calsequestrin and 𝗠𝐴𝐿-reticulin, the both are the calcium binding proteins. Now the second fellow is present only in smooth muscles. Okay, so these two I am adding here, important points. Now what if this Sarca pump is mutated? Okay, what if this Sarca pump is mutated? Then it's going to cause a disease called as Brody's myopathy. Okay, so Brody's myopathy. So in this video, what I have explained you, okay, I have explained you the differences between the contraction of the skeletal muscle, the electromechanical coupling versus electrochemical coupling. Electromechanical coupling is happening in the skeletal muscle, electrochemical coupling is happening in the cardiac myocyte. Okay, and I have explained you different types of channels which are present on the muscles, on the sarcolemma, on the T-tubules, on the L-tubules. Okay, different types of voltage gated sodium channels. Okay, different types of disorders, hyperkalemic periodic paralysis, hypokalemic periodic paralysis. Okay, Brody's myopathy. Okay, these things we have discussed. Now let me add one more important point here. Okay, this is skeletal muscle. Skeletal muscle, thing completed. In cardiac muscle, in the myocardium, you know there are two sources of calcium. Okay, there are two sources of calcium. Means one calcium coming into the cell from the extracellular fluid. Okay, and one calcium, other source of calcium, which is coming from the L-tubule from here. Okay, there are two sources of calcium. So the calcium which came from the sarcoplasmic reticulum, it's again going back. It's again going back with the help of Cerca pump. You know it. Now this calcium, whatever came from outside, it also again have to go back into the extracellular fluid. For that purpose, there is one more transporter present here. Okay, one more transporter present here. What this will do is, it will take the sodium into the cell and it will exchange the calcium out. It will exchange the calcium out of the cell. So the name of this transporter is called as sodium calcium exchanger. Okay, sodium calcium exchanger. Okay, so what is the importance of the sodium calcium exchanger? You will understand it in the pharmacology. Okay, in the pharmacology there are drugs called as cardiac glycosides like digitalis, digoxin. Okay, so if you use digitalis or digoxin, what happens? Okay, let me explain here itself so that you will having a better understanding. Okay, now look, let's say again, so this is the myocardium. Okay, so here is the L-tubule present. Here is the L-tubule present. Now the calcium from outside, this is the essential calcium which is coming into the cell. This is the essential calcium which came into the cell, and this is the calcium which came from the sarcoplasmic reticulum from here. Okay, now see on these cells, okay, on these cells, on the myocardium, there is this pump called as sodium potassium pump. It brings 2 potassium ions into the cell in exchange for 3 sodium. First 3 sodium will go out, so 2 potassium will come in with the help of ATP, by using the ATP. Okay, this is one channel present. Just now I have explained you, on this cell there is one more channel present. What this will do is, it's going to take sodium into the cell in exchange for the calcium, this calcium, the essential calcium, it will go out. Okay, this is called as a sodium calcium exchanger. Now look, if you use certain drugs like cardiac glycosides, digitalis, okay, digoxin. Now what this digoxin is going to do, it's going to inhibit the sodium potassium ATPases. When the sodium potassium ATPases are inhibited, okay, when the sodium potassium ATPases are inhibited, means what happens? See, normally this sodium, whatever is coming into the cell, sodium was going out. Okay, the sodium was going out with the help of sodium potassium ATPases. Now when you block this sodium potassium ATPases, do you think this sodium can go out? No, this sodium is not going out. Now do you think this calcium can go out? Okay, the calcium, this essential calcium, do you think it will go out easily? No. So it is increasing the intracellular calcium levels. So the drugs like cardiac glycosides, a digitalis group of drugs, digoxin, digitoxin group of drugs, they inhibit the sodium potassium ATP pump. If you inhibit the sodium potassium ATP pump, automatically the sodium calcium exchanger is also inhibited. So the calcium is not going out of the cell. When the calcium is not going out of the cell, the intracellular calcium levels increase. Now you know what this calcium is going to do. The intracellular calcium level, this essential calcium is not going out. It will stimulate the RYR channels to release more and more calcium. So more and more calcium is very much needed for the contraction of the heart. If more calcium is available means more powerful contraction. The heart will contract more powerfully. So that is called as the inotropic effect. Okay, that you will study in your pharmacology. Okay, so in this video I am just closing the video because this will be enough. We'll be doing one more part on how the actin myosin are going to interact, what are there in the sarcomere, what are the different types of regulatory proteins, what are the different types of anchoring proteins, what are the different types of contractile proteins and their disorders. We'll be discussing in the next video. But in this video, the differences between the cardiac muscle, myocardium, and also we have seen how it is different from the skeletal muscle contraction. Hope the video is helpful. Thank you.